Okay, well, let's get started. It is my great pleasure to welcome you all to this latest in our cyber series lectures, which invite members of our own Mount Sinai community to speak to us about their research. Today, I'm delighted to have Kristen Dams-O'Connor speak to us. It's been a great pleasure for me to work with Kristen over the years. She's currently director of the Brain Injury Research Center, and it's been very exciting to see her build Mount Sinai's efforts in the area of traumatic brain injury and related disorders. So Kristen, thank you so much for joining us. Please take it away.
Okay, I'm going to share my screen. Thank you for inviting me to give this talk. I really enjoyed these lectures, so I'm excited to be able to contribute now. Let's see, can you see a slide that is
Yes.
the advertisement for this talk?
Yeah.
Okay, let's see. Okay, so
yeah, perfect.
Can you see the title slide now?
Yes, perfect.
All right, I think we're good then. I'm gonna, let's see, I'm gonna actually stop my video just to make sure that there's adequate bandwidth here. All right, so the work that we do in our center really spans a lot of disciplines. It spans quite a few different types of research methods. So throughout this talk, I'm going to kind of go between zooming out to talk about some of the large data epidemiological research that we do, and then kind of zooming back into some of the smaller local studies, the more basic science, and kind of go back and forth between those big picture and then more zoomed in views, because that's kind of the way that our research is progressing. Each type of research is kind of informing our next steps. I'm grateful for the funding that is supporting this work. So I think, as many of you probably already know, the consequences of traumatic brain injury can range quite a bit. When I say traumatic brain injury, we're talking about an external force to the head or the neck that results in a loss or alteration of consciousness. So not a stroke, not an aneurysm, it's an external force. And in general, the more milder injuries tend to recover completely. And broadly speaking, people with more severe injuries are more likely to have lifelong deficits. And, you know, in this talk, I'm really going to be talking about the late effects of TBI. So the long-term effects of a remote TBI, which we know can differ enormously across individuals. We see patients clinically who have extremely severe injuries who are doing quite well a few years later, and individuals who don't make it out of the hospital, despite having more or less the same injury profile. The infographic that I'm going to pull up here comes from data in the TBI Model Systems National Database. So our group at Mount Sinai is one of 16 TBI model systems of care that are funded by the National Institute of Disability Independent Living and Rehabilitation Research. And these 16 centers across the country contribute data to what is now the largest prospective study of TBI outcomes in the world. So about 5 years ago, we partnered with the CDC to evaluate the representativeness of this database. And what you see here are a snapshot of the population weighted to represent individuals in the United States who receive inpatient rehab for TBI. So this largely encompasses people with moderate to severe TBI. And as you see here, the outcomes at 5 years post-injury are pretty grim. For many years, we have all been taught that traumatic brain injury is the strongest environmental risk factor for Alzheimer's disease. In broad strokes, I guess, the progression of that story is that many case studies kind of came out in the 70s to 80s presenting clinical cases of people with a severe brain injury who developed clinical Alzheimer's symptoms. And then in the next couple of decades, there were several epidemiological studies, cohort studies, that confirm this, or that is supported an association. And then studies of brain tissue out of the Glasgow Brain Bank from people who died acutely after TBI, so arguably before someone had a chance to develop a neurodegenerative disease, did still provide some support for this association when they found, for example, beta amyloid plaques that were similar in many ways to the plaques that are seen in Alzheimer's. I will say, though, that there really have not been many cases in the literature of people who were diagnosed with classic Alzheimer's disease during life. I'm talking about people with a traumatic brain injury, and then who were found to have really the hallmark neuropathological features of Alzheimer's disease upon death. So this association has been supported by a variety of research methods, but not across the continuum of clinical characterization to autopsy endpoints. I guess the methods around those research studies are kind of a separate talk, but I'll just say that a lot of those epidemiological studies have had some important limitations. One of which is a poorly characterized definition of post-traumatic neurodegeneration, post-traumatic dementia. So here we have a study that reports that the greatest risk, actually there are two studies that really got a lot of attention, but did the same thing. They found that the greatest risk for dementia after a traumatic brain injury is in the first 6 months. So both of these studies used claims data, and in all likelihood, what we're truly seeing here is the expected acute sequelae of a moderate to severe TBI. So this is clinicians who entered ICD-9 claims coding information, likely characterizing the acute cognitive changes after brain injury. And in fact, some of our colleagues recently did a review of the ICD-9 codes that were used in this paper and found that only 26% actually had any evidence of cognitive dysfunction in the medical record, and very few would meet clinical criteria for dementia. And given the timing of these findings, where the highest risk is in the first couple of months, it's extremely likely that those dementia cases, those dementia diagnoses predated the injury. So what is currently lacking in this area of research is a unified definition of post-traumatic dementia. A lot of studies are using the term dementia to refer to acute clinical sequelae, and they're also using all-cause dementia as kind of a catch-all term without specifying a dementia subtype. So when I talk about post-traumatic neurodegeneration, what I mean is kind of illustrated here. So this is a sample trajectory mapping an individual's pre-injury functional status. I made this myself, that's why it's not that pretty. I guess I should make a fancier one. But what you see here is the pre-injury functional status, whatever it is, and we know people start off being all different levels of function. Then you see a period of functional impairment. And of course, that also can vary in degree and in time course, followed by a period of recovery. So after that period of recovery, we know that people can have widely varying long-term trajectories after injury. So some do improve. We often forget about that. And I'm talking about many years after injury. A lot of people remain stable. I kind of talk about that in terms of chronic, static traumatic brain injury symptoms. And then others decline. And I think the important thing here is that decline, when we're talking about post-traumatic neurodegeneration, it needs to be defined by a change relative to a previously achieved post-injury level of function. It can't simply be defined in terms of pre-injury function. So we have used existing data resources, so large-scale data resources, to try to understand how common this really is. And this study used an approximation of that definition, so deterioration from a previously achieved post-injury outcome. And we see that functional decline is pretty common in people with a moderate to severe TBI. We also looked at this by age group, as you see here, and the short story is that age alone doesn't explain this decline. It's not just older people who are declining. We're obviously interested in exploring the association of traumatic brain injury and dementia in multiple different databases. So about 8 years ago, and this was actually around the time that the Institute of Medicine published their report about the long-term outcomes of traumatic brain injury, reporting with great confidence that traumatic brain injury is associated with increased risk for Alzheimer's disease. So what we did here is we used data from the Adult Changes in Thought study, a population-based longitudinal cognitive aging study based out of Seattle, Washington. And we looked at the association of TBI and dementia. And what you see here is that we found no association, not with Alzheimer's disease and not with all-cause dementia. But like I said, based on the knowledge at that time, this was a really unexpected finding. So we did a lot of sensitivity analyses. We looked at this a lot of different ways and didn't find an association. What we did next, about a year later, Paul Crane, who's the lead author of this paper, he's also the multiple PI of the Adult Changes in Thought study. We flew to Chicago to meet with colleagues at Rush University who lead the Memory and Aging Project and the Religious Order study. So two of the other largest longitudinal studies of cognitive aging in the country. And we pitched the idea of basically pooling the TBI and the dementia data across these three studies. The designs of the studies are similar enough that that made reasonable sense. They started around the same time. The inclusion criteria were quite similar. And what we found is no association between traumatic brain injury and the pathological hallmarks of Alzheimer's disease. So beta amyloid plaques, neurofibrillary tangles. We did find an association with Lewy bodies, which was kind of interesting. And even just from a power analysis perspective alone, we had a lot of pathology, a lot of, we had high Braak stage, frequent CR levels, and didn't find an association there. Lewy bodies were much less common, and still we found a reasonably strong association. We noticed that most of the people in these cohorts who had a TBI with loss of consciousness longer than an hour, so a severe injury, sustained their TBI before age 25. So we repeated these analyses looking at people with TBI with loss of consciousness younger than age 25. And as you see, we replicated those findings. So the Lewy body finding is interesting because clinically, and in some of the studies I'll share in a moment, we do find this apparent sort of motor subtype of clinical decline, where people many years after a brain injury seem to have, I should say, a subset of people. Not everyone declines, as I said before, but that would make sense pathologically as being a potential process underlying some of those observed clinical changes. I'll just skip ahead here. One of the limitations of those studies using the ACT database, ROS, MARS, is that they all used a traumatic brain injury ascertainment method that was based on a single item, asking people about a history of traumatic brain injury with loss of consciousness. And that, of course, prevents identification of injuries that are less severe. And also, based on the work we've done in our center, developing structured screening tools for lifetime history of traumatic brain injury, we know that those very broad single item questions tend to result in a lot of underreporting. And so what we did in the ACT study is we added the brain injury screening questionnaire, which we developed in our group around 2015. So we gave that some time to accumulate data. And they also did this incredible case review using the medical records available on this cohort. And incredibly, they had on average about 47 years of medical records for each participant in the study. And those records were meticulously abstracted. And so in what you see here, we used 3 different methods of TBI ascertainment. We used that really detailed structured questionnaire. We used the single item questionnaire that had been asked back starting in the 90s. And we used information from the medical records. So the reason we went to all this effort is because if you're misclassifying people with a traumatic brain injury into the no TBI group, you're obviously biasing things towards the null. And here what you see is no association, again, with the hallmark features of Alzheimer's disease. What we did find here, though, was an association with cortical atrophy. And I guess it's notable to say too that that association would not have been detected using only that single broad, single item TBI question. So I guess that fits with the idea that that an insensitive TBI exposure method results in some case misclassification and therefore obscure some of the potentially relevant associations. Now, I guess I'll zoom back in now. So those were all kind of large scale studies, big autopsy cohorts, but we know that a lot of the pathology that we have strong reason to believe would underlie a neurodegenerative process after traumatic brain injury is actually not even routinely studied in a standard neurodegenerative disease brain bank, like an ADRC type of a brain bank. So we didn't have large scale data available to us to look at things like inflammation, like axonal injury, blood-brain barrier disruption. And so we launched a study here at Mount Sinai with colleagues at the University of Washington and at Mass General, Spalding, Harvard, to begin a traumatic brain injury brain bank. I've talked about this a little bit before in some of the Friedman Brain Institute forums, so I'll just say this quickly. But what this is it's a prospective longitudinal study with autopsy endpoints. We're characterizing people during life longitudinally now with the funding that we just got last year. And everyone undergoes neurocognitive testing, neurological exam, structural and functional neuroimaging, blood draw for biomarker analyses, and then people who come to autopsy undergo this very detailed ex vivo protocol. I think I have a slide about that later, so I'll just move along here. But in designing this study, we tried to kind of learn from the limitation sections, I guess you could say, of all the papers that we had written using larger scale databases. I'll say just a few quick things about that. So how you measure traumatic brain injury exposure matters a lot. I think there's this conceptualization out there that even in these epi studies that medical records are the gold standard for lifetime TBI exposure, and it's just simply not true. We have found no study where where medical records alone provided a comprehensive lifetime exposure ascertainment. And when you think about it, that makes perfect sense. So a lot of injuries never make it into the medical record. They would need to be coded accurately for us to even pick them up in our algorithms. But all those injuries that don't, from people who don't seek care, who are injured in the context of military service, wouldn't make it into a civilian care system. People who are injured in domestic violence and an assault, those are notoriously underreported. And we even see this here at Mount Sinai on our rehab unit, that people sometimes make it a month after injury before the traumatic brain injury is diagnosed, because maybe they have a spinal cord injury or a crushed pelvis or other things that just simply supersede the traumatic brain injury in severity. And so it's not diagnosed or recognized until later in the course of care. So it's an excellent method, but it really needs to be triangulated with other methods. Self-report, I talked about that a little bit. How you ask matters a ton. And at some point in life, asking a significant other is nice complementary data, because in advanced age, you might begin to have concerns about recall bias. So that is something that we kept in mind as we designed our study, where we basically use all available information to characterize the injury and its acute severity. And then we thought a lot about selection bias too, which is a huge issue in these epidemiological studies. We know that there are things that can select a person into an injury. There are behavioral phenotypes that are associated with increased injury risk. Of course, sometimes the TBI is completely random, but people who select themselves into risk contexts are more likely to sustain injuries, and that has implications for how we understand the symptoms that develop after the injury. And I'll say, we also know that people on average who sustain a traumatic brain injury, especially in mid to older adulthood, tend to have more medical comorbidities before the injury. And that, of course, has implications for recovery as well. And then there's selection into a research study, right? So not everyone wants to be in a research study. We see this all the time in our center, where people on both ends of the severity spectrum are not as interested in research. So someone who recovers beautifully and is fully back living their pre-injury life, they don't identify as a person with brain injury as much anymore, or they don't have time to participate in research. And then similarly at the other end of the spectrum, someone who is living in an institutional setting is very unlikely to come into our community-based clinic and get in a scanner and participate in our study. So that's an important thing I think to keep in mind as well. And then there are things that contribute to retention in research studies. We've done some of this work looking at specific things associated with study dropout, study retention. And then of course there's selection into autopsy. So these are all the things that when we can, we measure them so that we can quantify them and then adjust for their impact on our findings. I'll say too that in prospective studies, we have found actually this is the work, a lot of the work that went into the development of the brain injury screening questionnaire, is we have used that measure now in a variety of settings. And we see alarmingly high rates of traumatic brain injury in certain settings that are systematically excluded from clinic-based research. So all those little bubbles are the proportion of people in a prison setting, in the homeless population. Those people around the table there, that's the general population. So some of our colleagues did a population-based study in Colorado and found that when you include mild traumatic brain injury, up to 40+% of people have sustained at least one injury in their lives. So when you're studying post-traumatic neurodegeneration, we started to really realize that we may be in some cases systematically excluding people who are at greatest risk for neurodegenerative decline and frankly the worst outcomes after an injury. So in the current late effects of TBI study, the LETBI study, we're actually working with colleagues here at Mount Sinai and in other community settings to specifically recruit from community resources that serve people who have been affected by intimate partner violence, the homeless community, the post-incarceration and parole communities, individuals who are receiving treatment for substance abuse. This is how we're recruiting now. And it's interesting because this stuff has been well known in the dementia literature for a while. So aging and dementia researchers have known for a while that clinic-based studies have different results than studies that are done in institutional and other settings. Like literally the association between risk factors and outcomes is different, sometimes in strength and also in direction, when you compare people in a given study who are recruited from a clinic to people who can only participate in that study if the study team goes to their home. So this is important stuff that I think is really often overlooked. TBD how feasible this is going to be. It's not going to be easy to follow some of these participants, but here's where we're at. So right now, this is the sample of our study. The point I want to make here, actually, it's better illustrated here. So a lot of the traumatic brain injury research happens in outpatient traumatic brain injury clinical settings. And a lot of it happens in the context of concussion studies in sports and in military service. So there's kind of two, I won't say silos, but kind of researchers, one of whom studies repetitive concussive and subconcussive head trauma in the context of sports and military service, and a different group of people oftentimes who are studying moderate to severe TBI. But what you see here is when you comprehensively characterize a person's head trauma exposure, they're not different people. So what you see here is that a lot of the people who have a single severe traumatic brain injury also have repetitive subconcussive head trauma. So when we're interpreting our findings, it's important to keep in mind that different exposure profiles may have different pathological processes underlying the clinical symptoms that we see, or they may not. That's actually one of the questions that our team is beginning to investigate along with others. So one of the kind of overarching goals of this study, of the LETBI study, is to achieve like really a multimodal characterization of post-traumatic neurodegeneration. We believe from our clinical exposure with these patients and the research we've done over the past 15 or so years is that some of the key clinical features are cognitive and neurobehavioral and motor symptoms. And we're interested to really understand the pathological mechanisms that underlie decline in each of those domains. So in the interest too of being able to diagnose post-traumatic neurodegeneration during life and ideally recognize some of the early signs, we are also attempting to identify plasma and imaging biomarkers that track with clinically significant change over time across one or more domains. And then, as I mentioned before, our plan is to use postmortem neuropathology to essentially validate those in vivo biomarkers. And this work really relies upon that network-based image-guided autopsy protocol that briefly, so just quickly, we scan the brains at high resolution postmortem to identify lesions in 3 dimensions, including those that in some cases were occult to standard neuropath methods. And then this information allows us to conduct an image-guided tissue selection approach so that in addition to conducting a standard autopsy workup on pre-selected brain regions, we can also investigate additional lesions that are potentially relevant to head trauma in particular. Those methods were initially developed with our colleagues at Mass General at the Martino Center. In fact, Bruce Fischel presented some of our data a couple years ago at the VIMI conference, I think when it was still TIMI. And we've now worked to implement that same protocol here at Mount Sinai. I'll say that Alan Seifert at VIMI has been instrumental in that effort. So we have begun to use the cross-sectional data that we've accumulated from our cohort to begin to try to understand whether there are distinct behavioral phenotypes. So we have now this really rich multidimensional assessment of cognition, of mood, of behavior symptoms, motor symptoms, and that kind of multidimensional data is obviously really well suited to phenotyping methods. So what you see here is we used a 2-step dimension reduction approach. So the first step was basically a factor analysis on those mixed data. And we then performed a hierarchical cluster analysis basically on the results of the factor analysis, so on the dimensions themselves. And then that cluster analysis, what you see here is 3 largely non-overlapping clusters. And the plot, what you see here is the plot of the individuals, how people in the cohort fall across those 3 clusters. And we placed people based on the first 2 dimension loadings, as you can see here on the slide. And more or less, what you see is a third of the sample, a third of the sample, a third. So there's some indication that we would be able to behaviorally subtype people with chronic, moderate to severe traumatic brain injury. And our hope now is, since we have MRI data on the sample, we're interested to begin to explore some of the neuroanatomical correlates of membership in these distinct behavioral phenotype clusters. But this kind of took us into the next challenge. I'll say that studying moderate to severe TBI is hard. It's really hard. So when we were generating hypotheses around the cortical networks that we thought would be associated with domain-specific clinical symptoms, I kept feeling very hesitant to specify a cortical network that I thought was going to be associated with decline in a given domain, because since we've been running this study, I've been seeing all the scans as they come in over the past 5 years. And these are people with remote, moderate to severe TBI. So in some cases, they have enormous lesions. So some of you on the call, you've been a part of the study, so you know that the radiologists who read the scans that are conducted for clinical or for research purposes, several times over the past several years, I've gotten calls saying, there's this, because they're looking for incidental findings, and the brains are alarming in some cases to look at. And so I'll get calls saying, oh my goodness, this is what we saw in this brain. But the findings of, enormous parts missing of brain is relatively expectable in this clinical sample. But it raises this question of how do you specify a cortical network when we know that many of these cases have large interruptions in those networks? But that approach is really standard in TBI research. So cortical volumetric analysis is widely used to study the anatomic basis of neurological deficits in traumatic brain injury. And the way that most of our colleagues have dealt with this large lesion problem is by excluding these patients. So if you look at a lot of the studies that are published, there'll be a sentence in there saying, we excluded people with large lesions, but that would be like half of our sample in this case. And so we spent a lot of time trying to think of ways to overcome this and in ways that would be broadly applicable to our growing study. And so what we did is develop a free surfer based lesion correction method. I mean, that's what you see here. That's the image here. So briefly, we reconstructed the cortical surfaces using T1 weighted MRI scans. And then Bram Diamond, who is a graduate student at Mass General, manually labeled them and removed the vertices of the cortical surfaces where the lesions caused inaccuracies. And then we remeasured the surface area of lesion overlap to calculate the percent volume of each network that was affected by a lesion. When we looked at this in aggregate, and I think at that time, about a year ago, we had about 87 cases to work with. And we found that the cortical lesions were preferentially located in the limbic and the default mode networks. And with those networks also having the largest area of surface volume affected. So what you see here is an illustration of the methods that I just went through. And I hope a reasonably convincing argument that this method has at least the potential to improve the accuracy of cortical volumetric measurements enough to allow us to permit inclusion in our study and studies like this of patients with lesioned brains. Of course, there would be a limit to what you can include. In some cases, we were just unable to perform this in a way that we felt was reasonable. In some cases, the lesions were just too large, too diffuse. But I think that broadly it provides some new opportunities to begin to understand network-based mechanisms underlying neurological deficits and their progression. So we've had a chance to apply this so far. Oh, this looks really weird. It's all stretched out, but you get the point, I think. So we looked at this over. This is just in a clinical study without imaging correlates, but we see obviously a range of functional change over time. Some people improve, some people stay the same, some decline. And then in the subset of people in the LETBI study who had undergone 2 scans at that point, and we just got funding to continue following this cohort about a year ago. So I think this is only about 30 people total. We do see some early indication, I think, that people who declined in motor function over about a 3-year period of time, there does seem to be some indication of cortical volume change in the associated motor network. So this work is ongoing. It's really pretty new. It's not enough people to publish yet. So we're kind of just beginning to explore how we can apply these methods to our growing database. Now I'll just say a little bit about CTE, because I know that is of great interest given the extent of media coverage it has received. let's see. So I recently had the opportunity to participate in a NIH funded consensus development effort to identify CTE criteria that could be applied during life. Now, I think that there are enormous caveats there. This was led by colleagues in Boston at BU, and what they did is they provided us with clinical information that was collected post-mortem from next of kin of individuals who had been diagnosed post-mortem with CTE. And we used those clinical data as early sort of criteria to develop what they're calling traumatic encephalopathy syndrome. I won't say a lot about the criteria because they're under peer review right now, but basically totally based on retrospective review. And the idea is to be able to diagnose CTE during life, which I think requires enormous caution and should not be done under any circumstance other than in a research context, but hopefully that will be clear in the published manuscript. So what we've done, because the goal, I mean, my interest in participating in that is to develop criteria that could be applied in existing research cohorts. So we've now made an attempt to apply those preliminary diagnostic criteria for traumatic encephalopathy syndrome to our LETBI sample. Now, again, these are just some of the basic features of our sample, and I'm going to skip ahead here just to the proportion of people across the severity groups that would meet traumatic encephalopathy syndrome criteria based on the data we have in the LETBI study. What you'll notice here is that the top row, that requires that an individual sustained repetitive head injury, but excludes people with a single traumatic brain injury. That is currently part of the criteria that are under peer review. This was an enormous piece of contention during the consensus process because we know that clinically the behavioral sequelae of a traumatic brain injury look a lot like what people are now calling traumatic encephalopathy syndrome. What we don't know is whether the underlying neuropathology is the same. But there is concern that if you diagnose, if a person would receive a different diagnosis for the same exact symptoms while making assumptions about the underlying pathology, then it may make it actually even harder to identify treatments that can be employed during life. So as you see, a large proportion of people with a moderate and severe traumatic brain injury, absent repetitive head injury, do meet the clinical criteria for traumatic encephalopathy syndrome. I'll say just a little bit now about some of the postmortem work that we've been able to do using our growing brain bank and also those of some of our colleagues. We, the LETBI team and John Crary, who's here from Mount Sinai, were able to participate in an NIH-funded consensus meeting to develop diagnostic criteria for CTE. Actually, new revised criteria. They aren't too different, but have some subtle differences, should be coming out pretty soon. But what we've begun to do is to try to address the question of whether CTE neuropathology is ever present in the context of single moderate to severe traumatic brain injury absent repetitive head trauma. What you see here, so we've begun to basically apply those consensus-based CTE diagnostic criteria in existing autopsy cohorts. So this comes from the adult changes in thought autopsy cohort. We selected a consecutive series of 200 cases from that cohort and applied those consensus criteria. And what you see is we found 3 cases out of 532 with the pathognomonic lesions of CTE. The rows A, B, and C represent cases with the pathognomonic lesions. They're blown up, you see, in the second column. And D, E, and F are cases that were suspicious for CTE, but did not have the pathognomonic lesions. Now, I think we have several pathologists on the call. The differences are subtle, so it is challenging to differentially diagnose CTE from subtle age-related changes from RTAG. So this too is an evolving field, but the point here is that we don't see a lot of CTE neuropathology in unselected community-based cohorts. We also had the chance to apply these same CTE diagnostic criteria to a series of, again, 200 cases from the University of Washington Alzheimer's Disease Research Center autopsy cohort. And the story is similar. We see very few cases of CTE, and we see that all of them are in males, and we see that only some of them are in cases with traumatic brain injury or with repetitive head injury exposure. In this study, because again, this is a study where people are characterized during life and then followed to autopsy, we did have the chance to look at associations with clinical information that was collected during life. And we did find an association between CTE neuropathology and motor impairment during life. And that's based on the UPDRS motor subscale score. So again, across a lot of these different studies and these different samples using really very different methods, we are seeing a story for motor change being associated with, so far, really two different types of two different processes, two different potential underlying pathologies. And then I won't spend a lot of time on this because what we've been able to do so far in our LETBI brain bank, which is accumulating cases, but still quite small. It still is a relatively small brain collection. I think it is the largest brain collection, to my knowledge, of cases who've survived many years after a single or multiple moderate to severe TBIs, but it still is quite small. So we are nowhere near the place of being able to do these large aggregate group analyses. And so at this point, we're really just doing a series of case studies. And it's fascinating. Each case is its own entire story, but I'll just give a couple examples here. So this is an individual who died at the age of 72, was diagnosed during life with atypical Alzheimer's disease and behavioral variant frontotemporal dementia. And we see in their brain, I mean, up in the corner there, that brain does not look good. There's a lot of atrophy there. And when we characterize, we speak to the families also. In addition to collecting data during life, we speak to the families postmortem to get another really complementary source of data. And this person had no known exposure to blast, although he did serve in the military, no exposure to combat, did not participate in contact sports. So to our knowledge, no repetitive head trauma, no subconcussive head trauma. There was a severe traumatic brain injury at age 37. Cognitive decline began around age 50, diagnosed with dementia at age 60. And what we found in this brain was some beta amyloid, not a lot, severe levels of p-tau across almost every single region examined. So low-level Alzheimer's disease, neuropathological change, a lot of tau, but we found evidence of PART, RTAG, no CTE. In this case, there was no evidence of those clear pathognomonic CTE lesions, no TDP-43, no vascular pathology. We did see quite a bit of astrogliosis, quite a bit of microgliosis. But here, broadly, this would be consistent with the idea that a single moderate to severe TBI is a mixed pathology, and that here we see no repetitive head trauma, no CTE. In this case, this is someone who had quite a bit of repetitive head trauma exposure. We think probably at least one blast exposure. The person served in the Navy. Serious traumatic brain injury in his 20s, and then another traumatic brain injury at age 47, decline beginning shortly thereafter, diagnosed in this case with atypical Alzheimer's disease. Here we see quite a bit of Alzheimer's disease neuropath changes. We see, I think the CRAD score was, I think it was C, very high, rock stage. Here we see no PART, no RTAG, but yes, CTE. You can see that quite clearly here on the lower left hand image. A lot of vascular pathology. And again, a lot of astrogliosis and microgliosis. And so again, like these cases are just kind of slowly accumulating. They, as you can see from the variety of things that we're describing, each case takes a lot of time and energy to characterize. But as these cases accumulate and as we're able to study them with the, I think, intensity that they deserve, we're beginning to piece together new information. Broadly speaking, in community-based unselected cohorts, we do not see very much CTE. We see some, but it's far less pronounced. You can see this here. The bottom two images are from the adult changes in thought study. The top is from a very high burden CTE case. So we, in community cohorts, we see that it's far less pronounced. And as I shared with the from the consecutive case series, its relationship with head trauma is much less clear. And then in our clinical samples, which, for example, in the LETBI study are limited to people with head trauma, when we look for traumatic encephalopathy syndrome across exposure subgroups, we have reason to believe that many people with only a single TBI would meet TES criteria. So it's very common in traumatic brain injury, but at least in our very preliminary investigations. And again, CTE itself seems to be quite rare in community samples. So we're, over time, I think, beginning to triangulate these multiple sources of information across multiple different methods. You will hear people say that CTE is only found in the context of repetitive head injury. So I guess from that perspective, if we're waiting for the exception that kind of disproves the rule, we have it. We have seen CTE pathognomonic lesions in cases with no known repetitive head trauma, and in fact, also in cases with no head trauma at all. I didn't share that here, but we have that from some other community-based studies. But as to the relationship between clinical symptoms and post-mortem neuropathology, we clearly have a lot to learn. This is why these various study designs, these different clinical cohorts with different selection criteria, with and those with autopsy endpoints are just so valuable to this work. And my hope, of course, is that as this cohort grows and as these studies mature, we will be able to learn more and more. So thank you for the chance to share this work. I think we have a few minutes for questions if anyone has any.
Kristen, that's great. Thank you so much for a wonderful talk. We do have time for questions. So please unshare so we can see one another. And I'll ask people to unmute themselves and ask Kristen any questions. Any questions from people?
Hi, Kristen. This is Andrew Varga.
Hi, Andrew. How are you?
Hi. Good. I guess I was just like a little bit confused. I mean, some of your early slides sort of seem to suggest that, you know, there was, right, the only sort of neurodegeneration biomarker that for which there was a strong association was actually, you know, alpha-synuclein, right, or Lewy bodies, but not so much for tau. I mean, but then, like, so clearly, people with one or more traumatic brain injury can have a fair amount of tau-associated lesions in the brain. So I don't know, how do I put that together?
Right. So in the study where we found the only association to be with Lewy bodies, actually in microinfarcts in that study, it had that very broad TBI ascertainment question, only asking about moderate to severe TBI, essentially, TBIs with loss of consciousness. And then when we looked back at that same cohort, pulling together multiple exposure methods, asking more detailed questions about lifetime history of traumatic brain injury, pulling in 40 plus years of medical records, we had much more detailed information. And a lot of the cases that went from the control group essentially then became TBI cases. And so when we were able to essentially improve case classification, we saw additional associations with cortical atrophy was what really popped up then. But your question is a good one. I mean, I am guessing you're thinking of it too from a perspective of sleep disorders, which we know to be extremely common following traumatic brain injury. And as we've discussed, another potential mechanism through which traumatic brain injury might initiate or maybe even accelerate neurodegenerative change. I didn't share it here, but we have been looking at just overall tau burden in traumatic brain injury cases as compared to controls in the adult changes in thought cohort. And it's sort of fascinating that we don't see huge differences. But one of the most enormous caveats, I think, of these large scale cognitive aging studies that have autopsy endpoints, and that's the adult changes in thought, the memory and aging project, religious order studies, all of those start at the age of 65 or greater. So you have to be at least 65 years to get into that study. You also have to be non-demented. So essentially you have to make it to advanced age, not demented, in order to get in the study. So anyone who dies prematurely after a traumatic brain injury, anyone who becomes demented early on is systematically excluded. And we know that mortality rates after traumatic brain injury, even among people who survive, are extremely high. Overall, the lifespan is shortened by 7 to 9 years among people who survive at least a year after a traumatic brain injury. We know that onset of clinical dementia is earlier in people with a traumatic brain injury. That all comes from epi studies, but you put the pieces together and it means that the people who are most likely to have serious neurodegenerative decline after a traumatic brain injury are again systematically excluded from those studies. So this is why it's so important to kind of pull together pieces from so many different studies with so many different methods and different selection criteria. Because do I believe it that people with a traumatic brain injury have no greater tau burden than people with? I don't. I think it's a selection factor.
Excellent. Other questions from folks?
Yes, this is Jim Ruda. Hi. On the myeloid end, you mentioned full recovery, but do you actually mean full recovery or like compensation?
So clinical recovery seems to happen in the fortunate 70 to 80% of people who seem to return to baseline. And your work studies a lot of the less fortunate people who don't seem to make that full recovery. But I mean, you've also seen, right, some subtle changes that persist even in those in the less severely injured cases. Is that right?
So like, well, yeah, but so like what I was asking was, when you say full recovery, is this that do you actually assume full recovery is possible, or you just say that the functional full recovery can be expected, but it occurs because of some compensatory mechanism done by other parts of the brain or like some other kind of learning?
Good question. So what I think we're beginning to understand is that even people, when I say full recovery, I mean no apparent clinical functional problem. But what we're actually seeing is probably the combination of some organic recovery combined with compensations that people are able to make in order to achieve that pre-injury level of function. But what we're beginning to understand, and I know your work has contributed to this, is that even in that subset of people who appear to have made a full recovery, there are still detectable pathological changes, especially if you're able to look using extremely sensitive tools.
Hi,
Thank you.
Kristen. This is Alita. Excellent presentation. The question that I have for you is sort of the visionary sort of question, which I'm excited to hear. Within the context of post-traumatic neurodegeneration, specifically moderate to severe traumatic brain injury, there are three sort of clinical outcomes that your work is looking to improve within the next, let's say, 30 years or so. What would you say are the most important things that your work would show would be helpful 3 years from now clinically?
Were you able to get that, Kristen?
I think so.
Your volume broke up quite a bit, but go ahead, Kristen.
I think I heard you, and if I don't answer it, please just tell me. So I think it was to do with what are the clinical sequelae that I would most like to be able to treat, that I think are highest priority to treat.
Right.
And I think my short answer is that I would like to treat the ones that are currently treatable with the methods we have available to us. So in our center, we also do clinical trials of neurobehavioral interventions, and we see success there. Those neurobehavioral interventions target the symptoms. They treat the symptoms. They don't treat the pathology, but until we are better able to understand the pathology and develop targeted treatments that can actually change the underlying disease course, we treat the symptoms. And we're pretty good at that. Neuropsychology, rehab psychology is pretty good at treating post-TBI symptoms. And I'll say that's one of the reasons too that I continue to rail against the any efforts to diagnose people with CTE during life. I think that there's a lot of concern, a lot of risk, and a lot of terrible outcomes that can come from telling a person that they have a neurodegenerative disease that they may not have. And until we're able to get better at that, we treat the symptoms.
Thank you.
Oh, I did think of one other thing. If I had to pick a clinical constellation of symptoms to treat, it would be emotional dysregulation, neurobehavioral outbursts. What we see, and it's interesting because a lot of people would think with traumatic brain injury that the most life-altering change is the cognitive change, and it certainly is. But the thing that unravels families, that unravels relationships, that results in loss of job, etc., is dysregulation, neurobehavioral, emotional discontrol, dysregulation. We have a trial going on in our center specifically targeting that. And I think that that's probably one of the highest priorities because, again, all of this work is in service of helping people who are living with brain injury. And in the near term, I think that that's where the movement is.
Kristen, do we know yet what form of tau accumulates with traumatic brain injury or chronic traumatic encephalopathy?
Very recently, and I would say that Claire and Susan and some others on the call may be able to discuss this in better detail. Very recently, there is some early evidence that it's slightly different than what is typically seen in Alzheimer's disease, that hyperphosphorylated tau. There is a subtle subtype that differs, but I don't know how well that has been accepted in the neuropathology community. I would, it's early findings, and I would honestly defer to my neuropath colleagues on that.
Yeah, it's just so fascinating to see the convergence of mechanisms between Alzheimer's disease and other dementias on the one hand, and traumatic brain injury on the other, just as you highlighted. And having a better understanding of the forms of tau that are induced in the different syndromes might be useful.
I think so too. And I think though that in the broader field of dementia research, the alignment between clinical symptoms and neuropathological processes is very imperfect. The clinical diagnoses that are rendered in life often do not match the pathological diagnoses that are rendered post-mortem. So that's something that I think we always have to keep in mind in our search for mechanisms underlying decline. If it's not well aligned in simpler processes, I don't know why we'd expect it to be any different in TBI. Right. It's hard. Yeah. It's a really hard thing to study.
Yeah, absolutely. I mean, having the brain bank that John and you and others are developing would obviously be a critical resource for that.
I hope so.
Well, we're just at the hour, so we should probably stop. Kristen, thank you again for a great talk. Thank you all for joining us. And if I don't see you all, please have a wonderful Thanksgiving.